Hi, this is the last of the biomolecules that we're going to be talking about. We're going to be talking about proteins for the next 8 or so lectures, and this will conclude the Chem 225 semester. So, proteins, as you know, are the most abundant class of biomolecules in our body. There are about 9,000 different kinds of proteins that exist in a cell. And then if you look at the whole human body, there's greater than 100,000 different kinds of proteins that exist in our body. So very, very important biomolecule to know about. So what we're going to do is, just like sugars and other biomolecules, we're going to define what these biomolecules are, and then go into depth about what make up these biomolecules. So what are proteins? Proteins are basically polymer, right? And polymer means that it's made up of monomers, and monomers are in protein amino acids. So is a natural polymer, not a man-made. Some of the examples of polymer are, basically your skin, your hair, everything that you think of, muscles, fingernails, silk, feather, all these things are basically natural polymers of amino acids. All right, so if you just approximately draw out what it would look like, let's say amino acid 1 is represented by A1, amino acid 2 is represented by A2, etc., etc. So these amino acids are linked through a bond, and these amino acids are linked by this bond that connects them, and this bond that connects them is an amide bond. All right, so just to briefly remind you what an amide bond looks like, is a C double bond O with a nitrogen attached to that. So this right here that I'm making it dark, that's what connects one amino acid to another. All right, so your book actually has a really good picture. I really like it because it shows in a pictorial form, in a puzzle piece. So they fit an amino acid inside a puzzle piece, and each of them fit together, and you make a big long puzzle piece, which would be a protein. And notice that they're all connected by this bond right here, in each case, which would be an amide bond. All right. So obviously, before we could talk about polymers called proteins, we need to first learn about the monomers, the individual amino acids that make up the protein. So we'll start there with the monomers, and then we'll go into how these amino acids come together to make proteins, and what properties they exhibit. All right. So as the name indicates, all right, amino acid. So let's look at the monomers. A lot of times these are called a residue, in a protein, and each of these residue or amino acid have two functional groups that are very important, and is indicated by the name. All right, so as the name indicates, you have the amino group, NH2, and you have the carboxylic acid functional group. All right, now there are a whole bunch of different amino acids that are present, that are available in nature. There are 20 of them that are abundant, and of the 20 amino acids, 10 of them are classified as essential amino acids because our body cannot make them. You have to get them through dietary source. But what is common about all these amino acids is that it has the amino group and the carboxylic acid group. And all of them will have what we call a side chain, which I'm going to indicate it by R, which means that it has a hydrogen on that carbon. So this alpha carbon right here, that is alpha to the carboxylic acid and alpha to the amino group, has R group called side chain. And that's how each amino acids vary by the identity of the R group, all right? So when R is H, then that amino acid is called glycine, which is the simplest amino acid you could have. This is not chiral. This is the only amino acid that's not chiral because the two hydrogens being same, all right? As long as R is not H, which is the case in all these other amino acids, so the 19 other ones, these are chiral, right? So it turns out that naturally occurring amino acids are L configuration. And what that means is that if you draw this in a Fisher projection and you have the highest oxidized carbon, which is carboxylic acid at the top, and you have the side chain down at the bottom, sort of like the way we drew for sugars, then it turns out that the amine group is on the left, which is why there are L amino acids. Remember, in sugar chemistry, the sugars were all D sugars, so the OHs were on the right. In amino acids, it's sort of the flip side, which is that the amino group is on the left side when drawn this way, all right? So if instead of doing the D or L, if you were assigning based on R versus S, most of the amino acids will give S configuration, all right? So that means that the nitrogen, carbon, and usually this is a carbon also, the nitrogen will go priority 1, and usually the carboxylic acid carbon gets a priority 2, and then the R group gets priority 3, and hydrogen's already going back, so you see that it gives you S configuration, okay? There is one exception to that, and that exception is an amino acid called cysteine, because the R has higher priority than the carboxylic acid, so it actually gives you an R configuration. That's why I say most. So 18 of these amino acids give you S, and one exception is cysteine, and that gives you an R, but it's still an L amino acid when you draw out in this Fisher projection. You have carboxylic acid here, amino on the left, and the CH2SH, the thiol group, is here, and just like other amino acids, all right? So what we've done to make things easier is to look at the 20 amino acids that's in this box, but they're divided into 4 categories, as you can see, all right? Now, in biochemistry, you're probably going to have to memorize all these amino acids, the name and what the side chain is. I'm not going to have you do that yet. You will do that eventually in biochemistry. I want you to focus on the chemistry of amino acids rather than spend your time memorizing these side chains. All right. So what I expect you to do is that I'm going to give you an R. So expectation. So given the side chain, right, the R group, it should be able to draw the amino acid. Okay, so for example, if I said alanine is the amino acid in which side chain is a methyl group, CH3, then you should be able to draw either in a Fisher projection or in a 3D what an each amino acid look like given the side chain. All right. The other thing I expect you to be able to do is to be able to categorize. Among these four groups. I'm going to go over that in a minute, but I just want you to know from the outset what you should be able to do. All right. So there are four categories the 20 amino acids are divided into, and given an R group, you should be able to tell me what category it belongs to. Okay. So the first category that we're going to look at is the non-polar amino acid. All right. So as the name indicates, if you look at the structures carefully, by the way, what is in the gray is the R group, the side chain. So notice everything else is all same. It's got the amino group and the carboxylic acid. So what's common about this all the amino acids that are in here is that you see a non-polar side chains. In other words, carbons and hydrogens, hydrophobic type of side chains. Okay, so all these belong to this. The thiol, it doesn't have a SH, it's a methyl thiol. This is a benzene group. Proline is a little different because the R group is inside the ring with the amine group as part of it, okay? And that's the only one that is that way of the amino acids. You might think that I'll come back to this tryptophan later on, ask when we're talking about other amino acids, why, even though it has a nitrogen in this side chain, why it doesn't belong to the basic amino acids. All right, so we'll come back to this later on. All right, so what is common about the non-polar amino acid is that they're hydrophobic in nature, and that should make sense. And a lot of these amino acids, because they're hydrophobic, they're found in the interior of proteins with limited interaction with water, right? And that should make sense, just like the how the phospholipids assemble themselves automatically. Same goes with these amino acids. If possible, they're going to be found in the interior of the protein, not in the exterior, and try to limit the interaction with water. All right. So this will conclude this lecture, and I'll continue with the other groups in the next one.